The method aids in the study of the mechanical signaling of the neurocrest cells and its interaction with chemical signals. It can also aid in the molecular and genetic mechanisms of the neurocrest development and diseases. Transfer the glass cover slips is the most challenging, therefore transfer them slowly and attentively to prevent breaking and dropping them. Demonstrating the procedure will be me and Dr. Sherry Zhao, a postdoctoral fellow from my laboratory.
Begin by placing the desired number of glass cover slips onto a piece of laboratory wipe. Then sterilize each cover slip by passing it back and forth through the flame of the alcohol burner. When the cover slips cool down, cover the 12 mm cover slip with approximately 200 microliters and 25 mm cover slip with 800 microliters of 0.1 molar sodium hydroxide. After 5 minutes, aspirate sodium hydroxide and allow the cover slips to air dry for 5 minutes to form an even film. Once the cover slips are dried, add approximately 80 microliters of aminopropyl triethoxysilane or APTS on a 12 mm cover slip and 150 microliters of APTS on a 25 mm cover slip. Aspirate excess APTS to allow the residual APTS to dry for 5 minutes. Later, rinse the cover slips thrice by submerging them in sterile, deionized water for 5 minutes each time.